Namaste to everyone. I hope all are fine and safe at home. Myself, Madhavi, I'll be taking biology to you all. So let us study about fundamental unit of life in this class. Introduction about the topic, that is the fundamental unit of life. What is this fundamental unit we are talking about? Let us know here. The cell is a Latin word meaning little room. So this is the slide where you can see the cells. Okay. When I say the cell is the fundamental unit of life, what am I telling about this fundamental unit is? The fundamental is forming a necessary base or core, which is of central importance, which is so important in life, which is forming a base. For example, for constructing a building, you need the base or the foundation to be very strong so that the building lies or lasts for long years. Otherwise, it will fall off if the base is weak, right? So, in the same way, the cell forms the basic unit which is of central importance. What is making it so important? Let us study in this chapter. Okay. So, as I said, the cell is a Latin word meaning little room. Here is the slide where you can see small rooms, right? It looks somewhat like a honeycomb like structures where honeybees keeps its honey inside the little compartments or little rooms. So it is similar to that of the cells, right? The honeycomb like structure is similar to the cells here. So the cell is called the little room. It has little compartments or little room. That is why it means little room. Robert Hooke, an English naturalist, discovered the cells in the year 1665. How did he discover? He took a slice of cork. Cork is nothing but the hard substance present in the bark of the tree. When he just scratched it and he took a piece of it and started observing under the microscope. Here, where I have written, he observed a thin slice of cork under a self designed microscope. Self designed in the sense he himself made his own microscope, since microscope was not so familiar in those days. It was not known much to the people. So, in those days, so he himself designed it and he started observing many things, under which he observed a slice of cork. He saw, or when he observed the cork, he could see these type of structures under the microscope. Okay, so it looked like similar to that of the honeycomb like structures which had little compartments. Okay, and the meaning is also called as little room. Microscope, what what is microscope? Microscope is an instrument used to observe micro objects that are completely invisible to our naked eyes. Micro objects, when I say, there are two things, micro and macro. Micro objects are those objects which are very minute and tiny objects which are not visible to our eyes. Okay, for example, bacteria, virus, many other microorganisms. We say microorganisms, that is why they are very tiny, which is not visible to our eyes. That is why microorganisms, organisms that are not visible, very tiny, very minute, they are called as microorganisms. And such organisms are seen under the microscope. And macro are the bigger objects. Say, for example, plants, animals, and many other things which are bigger, which our eyes can see it. Those are known as macro, micro, and macro. So microorganisms, very minute, tiny objects or the organisms are under an instrument called as microscope. Okay, microscope. Microscope was invented by Janssen Hans Lippershey. Janssen Hans, our father and son team together invented a microscope. And this microscope has a unit to express the size of the cell. That is, 1 micron is equal to 10 power minus 6 meter. That is, 1 micron is 1 millionth of meter. So this is expressed in 1 micron. And generally, there are two types of microscope, that is, compound microscope and electron microscope. Here is that this is compound microscope and this is electron microscope. Compound microscope magnifies the object about 2000 times, and electron microscope magnifies the object over 2 lakh times. That is, magnifies says that means it is giving larger view of the object. It is making the object appear bigger or larger so that our eyes can view it. So the compound microscope magnifies the object to 2000 times bigger than the actual size of the specimen. Whereas electron microscope gives 2 lakh times bigger or larger view of the specimen. So that is here you could see some cells and its structures, okay, whereas electron microscope can deeply give its structure, many other things present inside the specimen. So still more deeper details can be viewed using electron microscope compared to compound microscope, since there's difference in magnification. So these are the differences between compound microscope and electron microscope. And this compound microscope is used in laboratories, schools, colleges, and hospitals, etc. Whereas electron microscope is used in large scale laboratories, research centers by scientists and other things in order to do the research, because it gives inner details, and inner things can be studied under the electron microscope, uh, due to which, because it gives 2 lakh times the larger image of the larger view of the image object. So these are the main differences between compound microscope and electron microscope. I hope you have understood about this in this slide. This is the parts of the microscope. I will be explaining some main parts of it. These are the eyepiece. This is draw tube. This is the body of this body tube. This is the coarse adjustment. This is fine adjustment. This is the limb or the arm. Then this is the nose piece. This is objective. This is the stage. This is the slide. This rectangular thing is the slide. The circle thing is a specimen or the object which we are viewing. These are the clips to hold the slide properly. This is the condenser, this is mirror, this is foot, etc. So what is this eyepiece? Eyepiece is through which we see the specimen under the lens. That means our eye will be here. When we view through the eyes, we will first see here, so the eyes will be nearby, so that is why it is called as eyepiece. Okay, through which we see is, this is the place through which our eyes lies, or we see through this particular lens. So this eyepiece has eyepiece lens. Okay, and this is the body of it, and this is the objective. Objective has lens inside it. As we have eyepiece lens inside the eyepiece, here also we have lens. Okay, the objective lens, why it is called as objective lens? Because it is near the object. See, we keep the object here, right? So it is near the object, that is why it is called as objective lens. This is near the eye, that is why it is called as eyepiece. Okay, then stage. This is the stage to keep the slide. Something must be there, right? So this is the stage to keep the slide and the clips to hold it properly, so that we should get a proper view, right? In order to make it still, not to move here and there, we use this clip. In this slide, this round color one or the circle one is the specimen, what we are observing under the lens. Okay, and next is the course adjustment. What is this course adjustment? This is a rotating knob, okay, where we use to move forward and backward. What happens if we move this knob? Here, the lens keeps moving either towards the slide or away from the slide, in order to make the object more clear. We use this knob. Okay, the knob is used to move the objective lens. The objective lens here will be the lens, in order to move this near to the object or away in the sense, uh, away from the object is far from the object. Okay, either if we take sometimes far, it will be observed more clearly, or it is moved towards, so that we get a clear image, or I mean sorry, clear specimen view. Okay, so this is towards or away from the specimen being viewed. This fine adjustment is finally done with slight movement, so that the object or the specimen is um, becomes clear. In order to move in order to observe this particular object or the specimen placed here should be clear. That means we use this fine adjustment knob in order to make it. This we move only slightly, whereas this coarse adjustment we move the objective lens a little more, so that to get the clear image. Okay, for example, you can take when taking the photo in the camera, you keep using that zoom, right, to get a clear photo. So for the same purpose, we use this here as coarse adjustment and fine adjustment. Okay, then this is the mirror. What happens is, the in order to view the object or the specimen present inside the slide, we need light or brightness. Where will this brightness comes? When the light falls on the mirror, it reflects back, so that the slide will become brighter. When the slide becomes brighter, the specimen will be more clear to be visible to our eyes. So that is why the mirror is used here. The mirror projects or reflects the light, so the specimen is easier to see. Okay, these are main things which I have explained. Not much is there in your textbook. So now magnification. If the eyepiece lens, as I said, there will be one lens in the eyepiece also, and the lens will be there present on in the objective lens also, right? So this has some magnification like 10x, 20x, 40x, 100x, etc. Okay, that has a particular magnification. In the same way, even the objective lens has some magnification like 10x, 20x, 100x, 400x, etc. Okay, so based on the eyepiece magnification, we get here a bigger view. Okay, so say how to find out this. Say for example, if the eyepiece lens is 10x, if we are using 10x here, 10x lens, and the objective lens would be 40x, the total magnification we are getting will be, that means 10x into 40x, we get 400x. That is 400 times bigger it looks. Okay, that means the object appears 400 times larger than its actual size or the original size. The size may be very small, But using such magnifications, we get that much time bigger it looks, that much time larger it looks under the microscope. For example, the eyepiece may be 20x, okay, and here it may be 10x. So how much? 20x into 10x gives you 200x. So that much magnification or that much times bigger the specimen looks, okay, based on the eyepiece lens and also objective lens magnification. Our uh, getting the object clear would be deciding that only we use here that particular magnification, so that to make the specimen more clear, okay, and we get through these. That means here the object appears 400 times larger. It may be very small, but it looks 400 times bigger when viewed under the microscope. That is why the microscope is used in order to view very minute objects. Since they are not visible to our eyes, to make it visible to our eyes, we are using such type of lens inside this microscope, and we are able to see some tiny specimens, tiny organisms or tiny objects. Okay, these are some of the parts of microscope. Next slide. The cells and its types. The cells when viewed under the microscope. When viewed under the microscope, you can see here is thin lining, right? Thin black lining kind of, right? So the outline can be uh, of membrane. Since it is in the cell, it is called as cell membrane. It's thin outline, right? It is called as membrane or the cell membrane here. Here also you can see lightly it is visible. Okay, so these thin like outline is known as membrane, that is cell membrane. And the inner pinkish color, here also you can see inner some dotted or jelly like thing, that is cytoplasm. And here the middle dark purple color circle, dark, here also it is somewhat brown, okay, dark, something dark circle or dot like structure is known as nucleus. Okay, when viewed under the microscope you could see these basic structures or these basic parts. They are cell membrane, the cytoplasm and nucleus. Okay, the cells need not take the same shape or size of as I have shown here. It can take different shapes and sizes, as I have shown here types of cells. There are trillions of cells present in our body and in other microorganisms, I mean, in other organisms. One such types are here, mentioned here are muscle cells. See, they take the shape of this kind, and the fat cell look this, and the bone cells take the shape of this, blood cells, nervous cells, epithelial cells, etc. These are different shapes and the cells have different shape and sizes. Okay, it need not have always this, as I've shown in the slide. It can take different shapes. So there are some of the types which I have shown here. Okay, then uh, when I see these many things present inside the cell, there are when I if I say this is viewed under compound microscope. Okay, when I view one particular cell under electron microscope, this would look like this. Okay, can you imagine now what is the difference between compound and also the electron microscope? So one such cell in the compound microscope may look like this only basic parts. Whereas when observed under electron microscope, it gives a still more detailed study, or it gives a still more larger view, right? When viewed under one such cell, looks these many things, right? So the cell not only contains the cytoplasm, nucleus, cell membrane, it also has so many things inside, right, when viewed under electron microscope, right? So many things are there inside, not only these things. Now cell has special components As I said, membrane, cell membrane, cytoplasm inside, and also the nucleus. It also contains cell organelles. Okay, the cell organelles are, for example, mitochondria, endoplasmic reticulum, chloroplast, etc. All these are some of the cell organelles. What are these cell organelles? Let us study in the further sessions. Okay, so these cells has special components along with the cell organelles which perform specific functions or special functions. They perform functions. Each and every organelle do some its own work, which has made a cell a structure and functional unit of life. So all these together work and make the cells a functional unit of life. Okay, the cells contain special components and also the cell organelles which perform special functions that made or that has made a cell a structural and functional unit of life. Okay, let us study some more things in the next session. So thank you, children. Unicellular and multicellular organisms. Unicellular, when I say uni is single. We say unit, universal. Everything has uni here. So that is one, single. Okay, uni means single or one. Cellular in the sense which possess cell. So one cell, the organisms having one cell or a single cell are known as unicellular organisms. Multicellular, multi means many, more than one cell. Okay, the organisms having more than one cells or many cells are known as multicellular organisms. As I said, the unicellular, when an organism has to survive or has to live, it has to perform certain processes or functions. So here in unicellular and multicellular organisms, what happens is the processes like nutrition, digestion, excretion, etc., reproduction, etc., all these are performed by a single cell since there is only one cell. Unicellular organisms has one single cell which performs all the cell processes. It has to have nutrition. Nutrition in the sense it has to intake food, right? Nutrition is intake of food, right? Once it is taken inside, it has to get digested. The cell has to digest the food what it has taken, and the one which is digested has to be excreted. So that excretion, and the cell has to divide. So that is reproduction. So in that way, the processes are done by a single cell. Since there is only one cell, the one cell only performs all the processes. Okay, so unicellular organisms are those organisms having single cell and performs all the cell processes such as nutrition, digestion, excretion, etc. Okay, such organisms are called as unicellular organisms. For example, amoeba. See, it is intaking, here you can see it is taking the food inside, that is nutrition, taking the food inside. The inside material only is digesting and excreting. So only one cell, this is one cell performing all the activities of the cell. Here also paramecium, inside things are helping, right? So the one cell is only performing all the activities. Like that, there are many bacteria, chlamydomonas, planktons. When I say planktons, planktons are the water plants or animals. The plants or the animals that live in water. Tiny, tiny plants and tiny animals which are present in the water. The tiny or plants which are present in the water are known as phytoplanktons, and the tiny animals that are present in the water are known as zooplanktons. So in general, it is planktons. Okay, the tiny plants and animals that live in water. So these are the examples of unicellular. These have only one cell, and the one cell is possessing all the activities of the cell. Here comes the multicellular. As I said, multi is many. Many cells. There are many cells, and each cell does one function and together performs many functions. So organisms that have many cells that group together to perform cell processes. Like one cell performs nutrition, the other cell performs excretion, the other cell performs digestion, the other cell in reproduction. So in that way, they group of cells perform one function, group of cells perform another function. So in that way, each group of cells, there are many cells, and they make the group one set of group, and they perform nutrition. Okay, and the other set of cells perform another processes. So in that way, all the groups together help in the activities of the cell. Okay, since there are presence of many cells. Okay, this is the difference here. Only one cell is performing in the unicellular organisms, whereas multicellular organisms, there are presence of many cells, each group under one, and they perform their work. Okay, so such organisms are known as multicellular organisms, and they also form various body parts. Since they grouped under one and they are performing one such function, they become one of the part of it. Okay, so that, for example, fungi, plants, and animals. Okay, these are the multicellular. They have many cells inside which each one performs special or and specific functions. Okay, like for example, you can see if one person is there at home, a single person has to perform all the activities of the home. They have to do all the work of the home. A single person has to do when many people are there at home. So each one, uh, the work is being divided by each one, and each one does different functions. So that is the difference. Okay, that is similar to that of this unicellular and multicellular. Okay, so unicellular organisms and multicellular organisms has given the examples. Okay, thank you. Let us study some more things in the next session. Thank you, children.